Hello, my friends. We want to start. Do you hear me? Okay. Yesterday, I sent you the slide that you should study for your exam and lecture. Uh, all of them, I study you, I teaching you, but one of them I didn't teach. This slide did, okay, okay. This slide, do you see before or no? Do you see this before or no? Please study this malware. They are source of your exam. This slide, do you see before or did I teaching you or no? Okay. We start about today about this, okay? We want to speak about today about techniques of bacterial taxonomy. This is the classification of bacteria. We have three basic eukaryote, archaea, and bacteria. We want to speak today about this bacteria. And here we have some methods for bacterial taxonomy. It means for classification of bacteria, we need some methods. Here we have four famous methods. Microbial taxonomy is a science of study and grouping of microorganisms. Bacterial taxonomy concludes three separate but interrelated areas, three separate eukaryote, bacteria, and archaebacteria. For classification, we augmented organisms into groups, taxa, on the basis of similarities or relationship. For identification, process of characterizing organisms to determine an isolated as a member of an established taxon. Have next slide. Okay. This is important. How we taxonomy bacteria. For taxonomy bacteria, we have multiple methods, but three of them are very important for us. We can taxonomy or classification bacteria with their phenotype or genotype or phylogenetic analysis. This slide is important. Do you want writing or no? I continue. Okay. Come on here. This is more important. I called you three methods we have: phenotypic characters, genotypic characters, and phylogenetic analysis. Okay, what is phenotypic characters? Morphological data, physiological and biochemical data, and chemotaxonomic characters, such fatty acid analysis, the pattern of polar lipid present in the membrane, and composition of the cell wall. They are characters of phenotyping. And in genotyping characters, hybridization of DNA, guanine, and cytosine content, it means 50% guanine cytosine. And multicolor sequence typing, MLST, and DNA profiling. They are important in genotyping characters. And about phylogenetic analysis, we have 60 srRNA gene sequencing analysis, multi-gene sequence analysis, and whole genome sequence. Please write this slide. This is important for you. Please write in. Okay, do you writing? We have some specific concept in microbiology. It means no usually universally accept concept of species for prokaryote. Current definition of prokaryote species. Collection of strains sharing a high degree of similarity in several independent traits. Most important traits include 70% or greater DNA hybridization and 98.5% or greater 60 srRNA gene sequence identity. It is not important, okay? Don't study this slide, no. Okay, and here we have morphology. It is important. Phenotyping characters is important for classification. Morphology, what means? Microscopic morphology, we can classification with these characters. Cell morphology, rod, cocci, and spiridium. Cell agreement like Diplococcus or Streptococcus tetrad, Sarcina, irregular cluster, and a special cell structure cilia, spore, and capsule. Agil, please listen to me in this slide. I teaching you. I only take exam of until last slide. No more, no all of them. But until last section, please only listen to class. Yes, this slide is important. Please write it. Okay, do you writing? Okay, happening our next slide. This morphology, we explain about this to you. And we have colony morphology. Colonies can exhibit macroscopic difference like color or size, shape, margin or edge, and surface feature. And we have a slang culture morphology. This is important too. Please write this. Okay. About morphology, we have a staining. Okay, we have a gram stain. Uh, it is mean the reaction may later at the cell age and acidic staining, sodium black staining, or other methods for staining for a spore or capsule. Like this. Yes, this is important, I think. Okay. Do you hear me? Okay, okay. I don't want teaching you a lot today, but that slide I say you this important. Please pay attention and writing. And no more slide. Don't study. Okay, Hassan Fahim, don't worry. Okay. About physiological and biochemistry, I want to speak about phenotyping methods, okay? Phenotyping characters methods for classification. Physiological and biochemistry. The growth tolerance and enzyme activity, substance alterates antibiotic resistance. They are very important for physiology and biochemistry. And pay attention to test with identical media and conditions or at least comparable to compare the type strain of type species of operated genera and to analyze including a strain of the most closely related taxa than using the previously published data. Okay, have next slide. This is not important, not important, not important. Okay, here, it is important, writing, please writing. Antibiotic sensitivity. This is another phenotyping characters. The ability of microorganisms to withstand effect of an antibiotic on other pellets. Whether bacteria are susceptible, intermediate, or resistant depends on the amount of antibiotic. Here, this is sensitive, and this is intermediate, and this is susceptible. On the amount of antibiotic and the diameter of zone of inhibitor. Writing this slide is important. Writing. I explained you yesterday about exam. My exam is very simple and not very important, not very hard. Please don't worry. Only writing about this slide. Okay. What is serological analysis? Protein and polysaccharides of some bacteria can function as identity markers, generally molecules on surface structure like cell wall, galaxy, flagella, and pili. Detection is based upon this specific interaction between antibodies and this antigen, like this. Antigen and antibody. Writing this slide is important. Okay. It isn't important, don't study. And this. Okay. This slide is important. It means from slide 21, don't study until 32. Don't study. Do you understand? Okay. Importance of growth phenotypes. Can receptor of microbial, okay. Yes, start 21. Taxonomy. Bacterial identification, microbial ecology and evolution, cultivative more unknown bacteria. This is important. Writing. Okay, writing. Let's have new slides. In 1923, Bergy publishing a first edition of the term bacteriology, and it is based for classification and identification bacteria. It is not important. Okay. Chemical characters is in phenotyping characters, and it is important. Fatty acid analysis. What about speak about fatty acid analysis? Release on variation in type and proportion of fatty acid present in membrane lipids for specific prokaryote groups requires rigid standardization because frame profiles can vary as a function of temperature, growth phase, and growth media. Please write this. Please write. I continue. Okay. Okay. It isn't important. And these, please don't study. Okay? They are not important. Okay. This slide is important. Slide 53 is important. Writing. Okay. For genotyping analysis, we have several methods. DNA hybridization, one cytosine content, and multi-locus sequence typing, or DNA profiling. They are important. Writing. Okay. Yes, page 53. And this is important. DNA hybridization what means? It means a genome-wide compares of sequence similarly useful to distinguish species within a genus. Or in DNA profiling, we ribotyping AFLP or REPSIA or rapid method for distinguish between a species and a strain within species. And in MLST, strain typing using DNA sequences of multiple genes, high resolution, useful for distinguishing even very closely related strain within species. In GC or guanine cytosine ratio, percentage of guanine plus cytosine base pair in the genome, less commonly used in taxonomy because of poor resolution. If the guanine cytosine ratio of two organisms differs by more than about 5%, they cannot be closely related organisms with similar or even identical guanine cytosine relation may be incorrect. This is important. Okay, what is phylogenetic analysis? 60S RNA gene sequence analysis or multi-gene sequence analysis or whole genome sequence analysis. We use in phylogenetic analysis this method. Writing. Okay. I don't want spelling about this because I don't want your lecture to be more. Okay, I think this is enough for your exam. Which slide I explained today in this lecture, I take you exam. Which slide I didn't explain is not in your exam, is delete. Okay, do you understand? Do you understand? That's okay? That's okay? Okay. Don't worry about your exam. It is very, very easy. It is not important. I help you. I'm here and beside you. Please don't worry. Okay? If you have any question, please ask me. Next time, this is our last section in this class, okay? But next time we have class in microbiology, and we speak about only about enterobacterial cell. Okay, don't worry. If you have any question, I'm here. If not, bye.